Hi guys, welcome. We're going to be talking a little bit about biomolecules, so let's go ahead and start. All right, so biomolecules are organic molecules. These things always contain carbon. They're associated with living organisms. We have four types. They're carbohydrates, lipids, proteins, and nucleotides, right? Sometimes they're conjugated. Sometimes you have glycosylated molecules. They have both, a little bit of glycogen, protein, glycoproteins, and lipids. And polymers are made of repeating units, which are going to include things like glycogen, and you're going to see also amino acids coming together to form proteins. Well, first, let's talk a little bit about lipids, right? So notice that there's a carbon. Each one of these forms a bond, okay? That's actually a pair of electrons, okay, where you will see every little line is a pair of electrons. Here is a double bond, which means it's two pairs of electrons, right? This one's saturated. I mentioned it in class before. The saturated ones are going to have all carbons have as many hydrogens as possible. If they have double bonds with them, they cause kinks, and those kinks are going to create a mono unsaturated or poly unsaturated, because they have more than one, right? And then that means that they are going to be liquid at room temperature. All saturated fatty acids, like lard, what lard is made of, are going to be solid at room temperature. All of our lipids have a glycerol backbone, and they're going to have either 1, 2, or 3 fatty acids attached to them. So there's a lot of carbons involved in your lipids. They're going to be monoglycerides if there's 1, diglycerides if there's 2, triglycerides if there's 3. We have other lipid-related molecules, like the really important one is cholesterol, and phospholipids, eicosanoids as well. But definitely want to make sure that you know your cholesterol and your phospholipids. We're going to talk about phospholipids a little bit more, but we've already talked about them because we've talked about them when they are related to our cell membrane. All right, so then there's carbohydrates. So carbohydrates are going to have usually the 6 sugars. Yes, the 6 carbon sugars, excuse me. They are going to be fructose, glucose, galactose. Those are our 3 monosaccharides that are going to make up our carbohydrates. But we also have other sugars in other places. We have these 5 carbon sugars, which are going to be part of DNA, deoxyribose, and RNA, ribose. Okay? They are going to, these carbohydrates, monosaccharides are going to form disaccharides by making this bond right here. This bond is going to be created through something we call dehydration synthesis, which takes one of these OHs, one of these H's, and makes a water, okay? So then, for example, a glucose and a fructose together, they're going to create a sucrose. Glucose and glucose create a maltose. Galactose and glucose make a lactose. Then you're going to have chains, long chains of these glucose molecules. We make glycogen. Glycogen is something we store in our liver, for example, and in our muscles. Okay, now plants have a different type of long chain called cellulose, which we can't break down, right? They also make starch, which we can break down. So glycogen and starch we can break down, but we can't break down cellulose because we don't have the enzyme for that. That particular bond between this little O right here in cellulose is a little bit different. We can't break that one down. We can break down these. Okay, now proteins. Proteins are made up of amino acids. I want you to notice that there's a carbon in the middle. Yes, it makes 1, 2, 3, 4 single bonds with 4 different groups. This is called a carboxyl group. This one's an amino group. This one's going to be your hydrogen, and this R group here is going to differ depending on the amino acid. We have 20 different amino acids. So there's a bunch of different amino acids. Yes, usually you'll see them abbreviated by their 3-letter name. I really have rarely seen them in their 1-letter symbol. Right, so the primary structure of proteins is going to be the sequence of amino acids. Where does this sequence come from? This sequence, remember, comes from DNA. DNA is going to make an RNA, which is going to make a protein. And it's not all your DNA, it's a part of your DNA. We call it gene. A gene is going to make which is part of our DNA, make a RNA of that gene, and then we're going to make the protein from that RNA. Okay, so the sequence of amino acids, which comes from your messenger RNA, to be very specific, because we have 3 types of RNA, Your messenger RNA is going to give you your sequence of amino acids, right? The amino acids are then going to be in the form of nucleotides, right? You have go back. The sequence of amino acids is going to be the sequence of each one of these different amino acids. Okay, so amino acids have a primary, they also have a secondary, and they have a tertiary and quaternary structure. I'm sorry, those slides aren't on here, so I apologize for that. Okay, so the primary structure is the sequence of amino acids. The secondary structure is how those amino acids are going to be related to each other. They are going to form, they're going to be formed by hydrogen bonds. Remember, I went over that in class already. The hydrogen bonds are going to be between polarities, okay? So between polar molecules, so, or partially polar molecules. So we're going to have them keep that either alpha helix or beta pleated sheet shape that's kept by your hydrogen bonding. That starts to make these amino acids into three-dimensional structures. Then your long chain of proteins is going to make a tertiary structure, and that tertiary structure is going to be long alpha helices, which then move into a beta pleated sheet, which then move into an area of just a string of amino acids, which then combines to another alpha helix, and so on. That is the tertiary structure. However long your protein is, it's going to have multiple areas of different shapes, either alpha helix, beta pleated sheet, or something else, or not. Some little polarity here and there, but some hydrogen bonding, but not a lot. Then, for example, proteins can have a quaternary structure as well. The best example we have for that is hemoglobin, and hemoglobin has 4 different protein chains. So you have 4 different protein chains that make up one functional hemoglobin, right? So those are your 4 structures of protein. This one is talking only about primary structure, but you have your secondary, tertiary, quaternary structures as well. All right, and then nucleotides make up DNA. Okay, so DNA is going to be made up of a base, a phosphate group. Remember, phosphate groups are polar, right? And a sugar. So that makes DNA polar, and phosphate groups have RPO4 2 minus, which makes DNA negatively charged. DNA is negatively charged. The bases that we have are adenine, guanine. Notice that they are a double ring structure. We call a purine. Then you have the cytosine, thymine, and uracil, which are pyrimidines. They have a single ring. Thymine is found in your DNA. Uracil is found in RNA. So there are no T's in RNA, and there are no U's in DNA. So the way that it works is, for example, A's always bind with T's. Right? And C's always bind with G's. Okay? So in DNA. In RNA, we're going to replace that T with a U when we copy onto an RNA. We're going to talk more about this on Tuesday, because remember, I have that cute little lab for you on making DNA. Okay, so we'll talk a little bit more about that. But one quick little mnemonic that I use, your purines are pure as gold. PUR, A and G, so those are adenine and guanine, are your purines, and your pyrimidines are going to be cut the pi, and we use the letter Y instead of IE, so that you can remember that they are pyrimidines. Yes, so pure as gold for remembering that A and G are purines, and cut the pi, PY, so that you remember that cytosine, uracil, and thymine are pyrimidines. All right, so then this is your 5-carbon sugar back. Remember, I told you deoxy is missing an oxy. So notice here, yes, there's an oxygen here. The oxygen is missing. Why is it missing? Because we don't denote it. We don't denote hydrogen only. Remember, this here is a carbon. It has 1, 2, 3, 4 bonds. There's an H bond here, but we don't put it in if it's just a carbon-hydrogen bond. That is kind of the standard notation for organic chemistry. All right, that's your phosphate group, which I told you is PO4 2-. Okay? It likes to make bonds a lot of times, ionic bonds, remember, with calcium, because calcium is 2+, and phosphate is 2-. Okay, so here you go. Some of our nucleotides are going to be modified to be used for other things like, oh, I don't know, ATP. We're going to talk a lot about that next time when we talk a little bit about metabolism in chapter 3, 4, or 5. I believe it's, 4, 5, or 6. One of those. All right. So molecules and bonds. I'm going to go into, I actually talked a lot about molecules and bonding in class. So we're going to stop here. But that was a little introduction into some of your biomolecules, which is really important because biomolecules are obviously the foundation for all humans, right? Humans are made out of either carbohydrates, proteins, fats, and obviously DNA. Actually, everything in life is made out of those things, okay? So let's go ahead and stop here, and then I'll come back with a different little lecture.